CRISPR everywhere. Hi everyone, it's Dr. LeBlanc with a video capture lecture on CRISPR, Cas9, and genome editing. This is Nobel Prize winning science, ladies and gentlemen. Our learning goals are to understand what the function of the CRISPR-Cas9 system is in bacteria, to understand how the modified CRISPR-Cas9 system can knock out or replace genes with exquisite precision, and to understand how the CRISPR-Cas9 system can be engineered to target a variety of DNA and chromatin altering processes to precise locations in the genome. The CRISPR-Cas system is an antiviral defense mechanism that bacteria evolved to inactivate viral DNA. CRISPR stands for clustered, regularly interspersed short palindromic repeats, and that's what these diamonds are right here. This is a segment of the E. coli, bacteria, E. coli in this case, genome that has these short spacer regions that are interspersed. They're palindromic. And what are they interspersed between? Well, they are interspersed between spacer DNA that is derived from virus and integrated into this particular cassette in the bacterial genome. What happens when in the case of a viral attack? Well, an RNA is produced from these CRISPR regions, and it forms a hairpin because of the palindrome. This RNA binds to the Cas9 targeting DNA targeting machinery, which has endonuclease activity, and it also has helicase activity, and it can bind to the viral DNA and the endonucleases to chop it up. Now, there's a few more steps, of course, in this process that we've left out here, but these are the essential steps. When Jennifer Doudna and Emmanuel Charpentier discovered this in bacteria, they had great insight and realized what they had stumbled upon. They had found an enzyme system that could precisely target DNA sequences within a genome. And this is how it works. The Cas9 protein complex has a helicase activity that separates the two strands of DNA, and the guide RNA that is also bound to this Cas9 system can then hydrogen bond to a precise nucleotide sequence. In this case, it's the short interspersed sequences that have been captured from previous viral infections. However, in eukaryotes, this RNA can be replaced by RNA that can hydrogen bond to your gene of choice. So here's how the guide RNA and the Cas9 system works it can work in higher eukaryotes as well. This guide RNA is what makes the Cas9 system target specific sequences in the DNA. So if you remove the guide RNA that you found in E. coli and replace it with sequences that specifically target, say, a hemoglobin gene in a hematopoietic stem cell, you can add this guide RNA to the Cas9 system, and it has these cute little scissors here from Swiss Army knives that are going to cut the genome where the guide RNA hydrogen bonds to. Because the Cas9 has the helicase activity and the endonuclease activity, it can precisely position where in the genome is the Cas9 enzyme cuts the DNA. Okay, so here's another diagram showing the Cas9 system and the guide RNA hydrogen bonding to its target. The Cas9 enzyme cuts the target DNA here, and now what do you have? You have a double-stranded break. Well, there's two alternative mechanisms to fix this double-stranded break. One of them is non-homologous end joining. If you allow non-homologous end joining to occur, indel mutations result in the introduction of um, random numbers of nucleotides, that's the insertion, or deletion of random numbers of nucleotides. If you do that to your targeted DNA, you're going to end up with a frame shift mutation. And usually what happens when you get frame shift mutations is that you get premature stop codons being produced and you have effectively knocked out the gene. Very precisely knocked out the gene that you want to inactivate. So indel mutations will do that. If you allow non-homologous end joining to occur, you're going to end up with these indel mutations which result in premature stop codons. What if instead you want to replace this gene? Say there's a sickle cell mutation in there and you want to replace that sickle cell mutation with some DNA sequence that doesn't have that mutation in it. Well, if you supply at the same time homologous DNA, then the DNA repair mechanism for homologous recombination will occur, and you can end up swapping out the mutated DNA and inserting in its place, in precisely its place, the repaired segment of the DNA. So the CRISPR-Cas9 gene editing tools have been adapted from the prokaryotic immune system to provide sequence-specific genome targeting. That is the key feature of the CRISPR system. The Cas9 nuclease introduces the double-stranded breaks, 3 to 4 nucleotides, 5' from the PAM sequence. That's called a protospace or adjacent motif. It's not absolutely necessary to understand that part of the process right now. The break is repaired by one of two mechanisms: non-homologous end joining, which knocks out expression, or homologous recombination, which creates precise changes based on the template DNA. What if you want to do something a little bit more sophisticated? Well, you can hack CRISPR. There are the two main components of CRISPR are the Cas9 enzyme and the RNA that guides these molecular scissors to the sequence that the scientists want to cut out. Either cut and inactivate, or cut and homologous recombine, like we saw in the last slide. What if you alter the Cas9 protein? Okay, what if you break the scissors? Okay, if you genetically modify the Cas9 enzyme, you can still have the Cas9 enzyme bound to this region of the DNA, but if it doesn't cut, it's going to stick there. The Cas9 enzyme will block the binding of other proteins, such as RNA polymerase, needed to express a gene. What if you brought in an activator? Something that interacts with the mediator complex, for example. You could actually stimulate expression of a specific gene if you target this to the right sequence. A broken Cas9 enzyme can be coupled to epigenetic modifiers, such as those that add methyl groups to the DNA. If you add methyl groups to your DNA, you're going to most likely shut down the expression from this gene. On the other hand, you acetylate histones, you could activate a region of chromatin. Finally, you can actually make the CRISPR inducible so that Cas9, either dead or alive, could be coupled to switches so that it can be controlled by certain chemicals or as shown below by light. That way, you can have your Cas9 and guide RNA sitting in a cell waiting for a particular signal. That could be a light, a blue light, if you have a light-sensitive protein, and when the blue light is shown on the cell, you can activate the protein at precise times. Here are a few additional videos that you might want to consider watching. The Bozeman Science video is a surprisingly thorough introduction covering the bacterial origins of the CRISPR-Cas9 system in a little bit more detail in just 7 minutes. This is a very good video if you just watch the first 3 minutes and not the rest of it. The rest of it gets very complicated and talks about proprietary modifications to the Cas9 system. This particular video has a lot of very good animation and it's not too long. The first slide in this video was from the cover of Nature, and this has also been featured in the journal Nature a year or so ago. So in summary, the CRISPR-Cas9 system of precision genome editing was developed by Jennifer Doudna and Emmanuel Charpentier. Remember those names. They will be awarded the Nobel Prize in the not too distant future. From a bacterial defense mechanism against viral infection. The CRISPR-Cas9 system has both helicase activity and endonuclease activity to open a double-stranded DNA molecule and create a double-stranded break. In bacteria, the CRISPR-Cas9 uses a guide RNA to precisely target sequences in the viral genome for cutting. The CRISPR-Cas9 system has been bioengineered to use in precise gene targeting in eukaryotic cells. This has been accomplished by changing the sequence of the guide RNA to match the gene targeted in the genome. Genes can be knocked out by creating a double-stranded break in the middle of a gene coding region that is subsequently repaired by non-homologous end joining. This creates imprecise indel insertion and deletion mutations that create frame shifts. Alternatively, a copy of the targeted sequence can be supplied, and through homologous recombination, it can replace a gene, repair a mutation, for example. Many other applications of the CRISPR-Cas9 system can precisely target sequences in the genome in a way never before possible. And I should add that this technique can be used directly in cells in an organism or in cells in a Petri dish.